Hello, grade 8 students. In this video, we'll be summarizing the notes during semester 1. And here we go.
So let's start with part 1, immunology, chapter 1, the immune response, first activity about the self and
What is the immune system? The immune system, it's the army that protects our body against the harm around us like bacteria, viruses, parasites, and others.
so the immune system, let's start with the role of the immune system. What is the function of the immune system? What does it do? So the role of the immune system is to defend the body against any possible invasion by the bacteria, viruses, and parasites. So coronavirus is called COVID-19. So here you have to be careful about this. We're going to have a question similar to this, COVID-19. So the role of the immune system is to defend the body against any possible invasion by bacteria, by viruses, and so here immune system defense attacks citizens, foreigners. So as a conclusion, the immune system will defend or it will attack the citizens or the foreigners. So to start with, it defend that belong to my body. So my immune system defend my organs, my cells, my tissues. So the immune system will defend. We have two verbs, defend, attack. Okay? Defend or attack. So the immune system will defend the citizens, meaning that belong to my body, or the foreigners that are different from my body. Sure, the immune system will defend my citizens, my organs, my tissues. So defend is going to be for the citizens. So the immune system will defend the citizens and it will attack the foreigners. So the foreigners will be attacked by the immune system. The immune system will attack any non-self. So as a conclusion here, we have to say that the immune system defends its own citizens while it attacks any foreigners. So here is the conclusion, the first conclusion, and the very important conclusion here, that the immune system defends its own citizens, meaning that the immune system defends its organs, defends my tissue, my organs, my cells, while it will attack any foreigners. In the example, it will attack Ali's lung because Ali's lung is a foreigner with respect to Ahmed's body. So it's classified as a foreigner. So here, citizens, we're going to call them as self. Self, they belong to my body, to myself. While foreigners, they are classified as non-self. They don't belong to myself. They don't belong to my body. HLA. HLA, the definition, it's called the human leukocyte antigen. H for human, L for leukocyte, and A for antigen. So human leukocyte antigen, where it's found? It's found on the surface of nucleated cells. What the meaning of nucleated cells? Nucleated cells mean that they have nucleus. So what is HLA? HLA is the human leukocyte antigen. It's found on the surface of nucleated cells. It's found on the membrane of nucleated cells. What is the role of HLA? Why do we have to have HLA? HLA is the biological identity marker that helps the immune system to distinguish self from non-self. Let's say, for example, we have football club. Let's say we have Real Madrid and Barcelona. Okay, so Real Madrid and Barcelona, both of them, they have different logos. This is the logo for Real Madrid and this is the logo for Barcelona. So these two logos, they are different. How can we determine that this player belongs to Real Madrid on the field? By having the logo of Real Madrid and having the shirt of Real Madrid. So having the logo of Real Madrid, you can know that this player belongs to Real Madrid. Having the logo of Barcelona, you know that this player belongs to Barcelona. So HLA, it's like the logo. For example, having the logo on your shirt for ISI, you know that the student belongs to the International School of Innovation. So by this way, we can determine the student belongs to the ISI. Here, biological identity marker, this will help the immune system to distinguish the self from non-self. If this cell have the HLA, so it belongs to my body. So it's a self. So the immune system won't attack it. It will be safe. So the immune system depends on the HLA. So HLA is the biological identity marker that helps the immune system to distinguish the self from non-self. So if we have the HLA on the cell, meaning that it has the logo, it have the marker, it have the identity marker, okay? So this cell belongs to my body. Another example, you have the Lebanese ID, okay? So wherever you pass, you have this ID, meaning that you are Lebanese, so you can pass. If you don't have your ID, meaning that you are not Lebanese, you can't pass. So this one is called an example about the biological identity marker. So the biological identity marker, it helps the immune system to distinguish the self or non-self. If you have the HLA, meaning that we have the self cells. If they don't have the HLA, they are non-self. So, third note, the cells of the same body and those of identical twins, they have the same HLA. So for example, my heart, my kidney, my liver, my stomach, for example, my eyes, my skin, and others, they belong to my body. So they are the cells of the same body. So they have same HLA. Also, identical twins, meaning that identical twins, they look like each other. Okay? So identical twins, they have also the same HLA. While the cells of different persons, for example, the cells of Hassan and Ali, they don't belong to each other. They are, for example, friends only in the class, so they have different HLA. So as a revision, what is HLA? It's a human leukocyte antigen. It's found on the surface of the nucleated cells. It's the biological identity marker. It's the marker, and it's specific for the biological identity marker that helps the immune system to distinguish the self from non-self. So if it's self, it should have the same HLA. If it's not self, it should have different HLA. Move now. Some notes about the HLA. Each person has a specific HLA, on the surface of nucleated cells, the cells that have nucleus. But wait a moment, we have special types of cells. They don't have nucleus inside our body. What do we call them? They are the red blood cells. The RBCs, red blood cells. they don't have a nucleus. So they don't have HLA. That's why we can donate blood for any person. So we don't have HLA. So the immune system will not attack these red blood cells and will not consider them as non-self. Why? Because they are anucleated or anucleated, meaning that they don't have nucleus. So red blood cells, pay attention to this note, it's very important. Red blood cells, they don't have a nucleus. So they don't have HLA, because we said that HLA is found on the surface of nucleated cells. Also, identical twins, as I said before, they have identical HLA on their nucleated cells. So now, let's develop Freire's model for self and non-self using the HLA. Now, this is the complex definition for the self. Here, Freire's model, here we have the term in the middle, here we have the definition, here an example, here a non-example about them. Okay, so let's start first with the term self. What the meaning of self? We said before that self is all that belongs to one's body, like my hand, my kidney, my stomach, and others. Here we added a few terms or few words and has the same HLA. So whenever you ask about the term of the self, you have to say that all that belongs to one's body and they have the same HLA. Without same HLA, meaning that you don't know the definition of self. So self, all that belongs to one's body and they have the same HLA. Self, example, my hand, my kidney, my liver, my pancreas, my eye, my nose, and others. They are called self. Here, another example, my friend's hand. My friend's hand is not self. It belongs to others' body. It's called non-self. So as revision, what is the self? Self is all that belongs to one's body and has the same HLA. Example, my hand, my liver, my pancreas, my kidney, my stomach, my eye. Non-example, my friend's hand, my friend's kidney, my friend's nose. All of them, they are non-examples. Okay, let's move now to the definition of the non-self. So the non-self. What is the non-self? Definition. The non-self, all that does not belong to one's body. And here we have additional information here, and they have different HLA. So what the difference between self and non-self? We said that the self is all that belongs to one's body and they have the same HLA. While the non-self, all that does not belong to one's body and they has different HLA. Example about the non-self, my friend's hand, my friend's kidney, my friend's lung. Non-example, my hand. This is my hand. Example about the self. So here we have the definition of the self and non-self. You have to know them. Self, all that belongs to one's body and they have the same HLA. Non-self, all that does not belong to one's body and they have different HLA. Now let's move to an idea about grafting. What is a graft? A graft is a transplant of a tissue or an organ from one side to another in the same body or from one body to another it's moving from an organ. You know that some people, they can donate a kidney, for example, or they can donate a lung, or they can donate, for example, let's say an eye. So this is called grafting. So grafting is the transplant of a tissue or an organ from one side to another. For example, some people, they have, for example, problems in their face. They have, for example, burned their face, for example, by fire or something. So they have damaged skin cells. So they can take skin cells from the thigh, from the leg, and they can transplant it into their face to have the recovery for the skin. So this is from one side to another in the same body. Or, for example, we can transplant an organ from one body to another. For example, I can donate a kidney for my sister, or I can donate a liver, for example, or I can donate, let's say, a lung to my brother, let's say. So this is from one body to another. Here we have different types of grafts. Let's start with the first type. First type is called autograft. Auto, definition, is the graft that is transferred from one side to another in the same body. So it's transferred, the graft is transferred, or the transplant is transferred from one side, from one position to another position in the same body. Example about this, from my hand to my thigh, either from my hand to my leg, or from my thigh to my face, let's say. This is an example. Another example, from my hand to my friend's hand. This is a non-example. Okay? Another type of graft is called isograft. Isograft is done between a person and the identical twin. So always I, identical, I, iso. So I, identical twin, isograft. I, identical twin, isograft. Here, Samir's kidney transferred to his identical twin. Maybe it's called Fadi, for example. So isograft is the graft that is done between two identical twin. This is an example. Not an example, Samir's kidney transferred to his sister twin. Sister twin, pay attention. Sister twin is not an identical twin. So pay attention to this. They should be, both of them should be males, or both of them should be females in order to say that they are identical. So, identical twin, the graft that's done between two identical twins is called isograft. So till now, we have two different types of graft. The first graft is called autograft. The graft is transferred from one side of the body to another side in the same body. And we have the second type, isograft, the graft that is transferred from one person to the identical twin. That's why I, identical, I, iso. So I, identical, I, iso. So you can easily detect this. Let's move now to the third type of graft, called allograft. This graft, allograft, is done between one person and another in the same species, and they belong to the same species. For example, from Hassan to Maya, from Ali to Riwa, from Celine to Sabine, from Sabine to Zahra. So all of them, they belong to the same species. Okay? Here, for example, the graft is transferred from one person to another in the same species. Okay? So this is called allograft. Here example about this, from Maya to Rola, from Hassan to Hassan, from Kareem to Hadi, from Hadi to Rida, from Rida to Abbas, from Celine to, let's say, to Fatima. All of them, they belong to the same species. While an example, from a cat to a dog. Cat and dog, they belong to different species. So it's not an example about allograft. graft is called xenograft. Xenograft is the graft that's done between two different individuals that belong to different species. For example, from a cat to a dog, from a dog to a horse, from a horse to, for example, let's say zebra, from zebra to lion. Okay, so they belong to different species. From, for example, from a human to a dog, let's say, or from a dog to a human, they belong to different species. So this is an example. From a cat to a dog, cat and dog, they belong to different species. Cat and, for example, a hen, let's say. So they are different species. So here we have 4 types of graft. Autograft, within the same body. Auto, within the same body. Iso, iso, identical. Allo, different individuals in the same species, from a human to a human, from a cat to a cat, from a dog to a dog, from, for example, a horse to another horse. They belong to the same species. While xenograft, they belong to different species, from a cat to a dog, from a dog to a hen, let's say, from a dog to a human. So they belong to different species.
So what affects the fate of the graft? The fate of a graft, it depends on the similarity of HLA between the donor and the recipient. The donor is the one that donates or gives, and the recipient the one that receives or takes. So the fate of the graft, whether it's successful one or it failed, it depends on the similarity of the HLA. Now, let's start with the blood groups, or what's called by the ABO system. ABO blood grouping. They are, according to the ABO blood grouping, there are 4 different kinds. We have blood group A, blood group B, blood group AB, and blood group O. Blood group A, if you have blood group A, so you have antigen A and you have antibody B in your blood plasma. So if you belong to blood group A, you have antigen A on the surface of your red blood cells and you have antibody B. While if you are blood group B, then you have B antigens on the surface of the red blood cells, so you have antigen B and antibody A in your blood plasma. So blood group A, as you notice, the antigens, they are similar to the blood group. So blood group A, we have antigen A, and the opposite of the antigen A is antibody B. While blood group B, we have antigens B and antibody A. Okay? None of the antigens and its corresponding antibody should be present in the same blood group, since if we have antigen A and antibody A within the same group, so we will have agglutination. Blood group AB, you have both antigens, A and B antigens, on the surface of your red blood cells, and there is no anti-A and no anti-B antibodies. Blood group O, it's called the null group, neither antigen A or antigen B, so no antigens A and B, but we have both antibodies in the plasma, both the antibody A and antibody B. Many people also have a so-called Rh factor on the red blood cells. This is also an antigen, and those who have it are called Rh+. That's why you have, for example, A positive, B positive, O positive, B positive. Those who won't have it are called Rh negative, meaning that Rh minus, like A minus, A negative, B negative, AB negative, or O negative. So a person with Rh minus blood does not have Rh antibodies naturally in the blood plasma. But a person with Rh minus can develop Rh antibodies, or she receive blood if he or she receive blood from Rh plus blood. A person with Rh plus blood can receive blood from a person with Rh minus without any problem. So this table showing the different blood groups there are different antigens, antibodies for whom they can donate, from whom they can receive. So blood group AB, it has both antigens A and B. It has no antibodies, can give only to the AB, but can receive from all the blood groups AB, and O. So AB is called the universal recipient since it receives blood from the different blood groups. Whereas blood group A, it has antigen A and antibody B, can give blood to A and AB, and can receive from only A and O. Blood group B, it has antigen B, it has antibody A, and can give blood to blood group B and AB, and receive from B and O. Blood group O, it has no antigens, while it has both antibodies A and B, can give blood to A, B, and O, and can receive from O only. That's why the group O is called the universal donor, since it can donate for the all blood groups. Let's move now to the different immune cells in the chapter of immunity. First, let's talk about the blood cells. Blood cells, they are divided either into red blood cells or white blood cells. Red blood cells, they are called the erythrocytes, whereas the white blood cells, they are called the leukocytes. Red blood cells, they are small in size, many in number, whereas the white blood cells, they are large in size and few in number. They are red in color, the red blood cells, while they are not red in color. They don't have red color, and they are not white. They are anucleated, the red blood cells, meaning that they don't have nucleus, whereas white blood cells, they are nucleated, they have nucleus. The role of the red blood cells is to transport oxygen and carbon dioxide, while the role of the white blood cells is to defend the body, or they have a role in immunity, immune cells. Cells involved in immunity. Monocytes. They are the cells involved in immunity. What do they have? They have horseshoe-shaped nucleus. They are large in size, flow in the blood vessels, can cross the capillary walls, mature into macrophage, and have a role in phagocytosis. So we can determine them through their horseshoe-shaped nucleus. Other type of cells is called granulocyte. What is special about the granulocyte? They have multi-lobed nucleus. So they are large in size, they have multi-lobed nucleus, they have granules in their cytoplasm, they can flow in the blood vessels, they can cross the capillary walls, and they have a role in phagocytosis. B lymphocytes. We have two types of lymphocytes, B and T. B lymphocytes, they are large in size, have large rounded nucleus, small cytoplasm. They have a role in secretion of antibodies to kill the non-cell. These are the antibodies. Okay, this is the figure showing the red blood cells, the B lymphocytes. They have large rounded nucleus, small cytoplasm, cytoplasmic membrane, and the antibodies found. T lymphocytes. T lymphocytes, they are large in size, have large rounded nucleus, have small cytoplasm, and they are called killer cells. They use their TCR, T cell receptors, to kill the non-self. Also, they have small cytoplasm, large rounded nucleus, and they have TCR, T cell receptor. Immune organs, they are divided either into primary or secondary immune organs. Primary immune organs, they are the organs of production and the maturation of the immune cells, whereas secondary immune organs, they are the sites of the immune response. Immune organs, they are divided into either primary immune organs or secondary immune organs. Primary immune organs include the bone marrow and the thymus, while the secondary immune organs, they include the lymph nodes and the spleen. What is the role of the bone marrow? Production of all the immune cells, production of B and T, but it only matures B lymphocytes. Thymus has only one role, maturation of the T lymphocytes. Secondary immune organs, as we said, is the site where the immune response takes place. Lymph nodes, immune responses against tissue infection, whereas spleen, immune responses against blood infection. Let's move now to the natural barriers. Natural barriers will form the first line of defense inside the body. So what would happen if these natural barriers were broken? If the first line of defense is broken, we have infection. So infection is the invasion and the multiplication of pathogens in the. If the first line of defense is broken, we have a pathogen. What is the pathogen? It's any non-self organism that's found in the body that infects the body, like the case of bacteria, viruses, and others, and it's capable of causing a disease. Inflammatory response. What is inflammation? Inflammation is the immune response against infection. It's the second line of defense in our body. Inflammatory response includes four signs. First one, redness. Second, heat. Third, swelling, which is called edema. And finally, we have pain. These are the four signs of inflammation. Note that the first cells to cross the capillary walls, they are the monocytes and the granulocytes. That's why they are called phagocytes. Characteristics of inflammation. It's instantaneous and non-specific. What the meaning of instantaneous? It starts immediately. Non-specific meaning that it does not depend on the identity of the pathogen. Phagocytosis. What are the steps? First, the phagocyte approaches to the pathogen. Second step, the phagocyte adheres to the pathogen and gets indirect contact with it. Number 3, the phagocyte engulfs, ingests, and digests the pathogen. Main actors of the phagocytosis, they are the phagocytes. What are they? They are monocyte and granulocyte. Phagocytosis is the non-specific immediate immune response. It starts immediately against any infection. The main actors, they are the phagocytes, who are monocytes and granulocytes. Let's talk about the specific immune response. Specific immune response, first line of defense, it represents the natural barriers. Second line of defense, the non-specific immune response. What will happen when the second line of defense fails? We will have the third line of defense, which is a specific immune response. What are the steps of the activation of T lymphocytes? We have, number 1, the specific T lymphocytes. They approach the target or infected cell. Second, the T lymphocytes adheres or bind to the target infected cell using its TCR, T cell receptor. Third step, the T lymphocytes can give the target cell a killer kiss. It perforates, creates holes in it. As such, the target cell dies. What is the type of the immune response carried out by T lymphocytes? Sure, it's specific immune response. The name of this response is called cell-mediated immune response. So we can conclude that T lymphocytes, they carry out specific cell-mediated immune response, where T lymphocytes, they kill the cells in both. Type of the immune response carried out by B lymphocytes is called specific immune response. The name is called humoral immune response. So B lymphocytes, they carry what is known by specific humoral immune response, where B lymphocytes, they secrete antibodies against the non-self into the serum of both cases. Let's talk about the characteristics of the specific immune response. You have to keep in mind here that the primary immune response is the immune response upon the first contact with the pathogen. So we call it primary immune response. Whereas the secondary immune response is the immune response upon the second contact But pay attention here with the same pathogen. You have to keep in mind also that multiplication, it means proliferation, it means amplification. Antibody secreting cells, they are called plasma cells. During differentiation, a cell differentiates into either effector cell, that will be B cells or T cells, or memory cells that has an important role. Effector B cells, they are called the plasma cells, whereas effector T cells, they are called the killer cells. So this is the primary immune response upon the first injection of the antigen, and this is the secondary immune response upon the second injection of the same antigen. So long latency in the primary, it takes about 10 days, while short latency in the secondary immune response, it takes about 2 days. While low amount of antibodies in the case of primary and high amount of antibodies in the case of the secondary immune response. Short duration of protection during the primary immune response, while long duration of protection during the secondary immune response. So latency and this table showing the comparison between the primary and the secondary immune response. It shows the latency, which is the time needed to start the immune response, amount of antibodies, and duration of the response. Latency is the time needed to start the immune response, where it's long in the case of primary and it's short in the case of the secondary. Low amount of antibodies in the case of the primary, high amount of antibodies in the case of the secondary. Short duration of response in the case of the primary, and longer duration in the case of the secondary immune response. Also, you have to keep in mind that primary immune response takes a longer time to eliminate the infection, but it produces memory B and T cells. They are used for subsequent invasions. Secondary immune response takes a shorter time and is more efficient. Why? Due to the presence of these memory cells from the primary immune response.